Thank you. Thanks for the opportunity to present today. It seems like a great group who I'd like to get to know better over the next couple of years. So um, I'll start with a little bit of background about me and then tell you about some of the work that my lab has been done focusing on the evolution of the human gut microbiome. So I'm an evolutionary biologist. My work is motivated primarily by the observation that no species evolves in isolation. So every species on Earth is deeply embedded in this web of interactions with other species that can range from antagonistic interactions like predator-prey interactions all the way towards mutually beneficial interactions. If you were to open an ecology textbook, you may see a slide like this showing species interactions between macroscopic organisms, but the vast majority of interactions that affect us and our health, I would argue, look something more like this. So this is a scanning electron microscope image of a microbial community assembling on a food particle in the human gut. This is a falsely colored image. It's actually pitch black in your gut, so it doesn't look like this, so you all know. The different colors here outline different bacterial taxa. So within each of us, there's about 30 trillion cells, bacterial cells, numbering roughly the same as this number of cells in your body, the human cells in your body. So each of us now, as you're eating lunch, are feeding these multitudes of gut bacteria that we know from experimental work in mouse models and also work in humans are deeply integrated with various phenotypes and fitness in humans. So we rely, the microbiota relies on us, but we also rely on the microbiota for our own development. So the microbiota is essential not only for the development of the digestive system, but it's also involved in training and the development of the circulatory system, the immune system, myriad other tissue and organ systems throughout the body. And yeah, even has effects on our neuroendocrine system that control our moods and behaviors. When I started as a grad student in 2010, about 15 years ago, there were many studies emerging showing these relationships between the microbiota and host phenotypes, but we didn't have a very good understanding of how these symbioses have evolved. And so this is the big question that I've been working on for the past 15 years, and my lab continues to work on it. I will hopefully give you a partial answer to this question today, focusing on humans. So in the first part of the talk, I want to tell you about some work we've done looking at how the microbiome has changed over the course of human evolution, going back to our ancestors with African apes, and then moving forward in time through human evolution, and even to the very recent time with the rise of industrialization and various lifestyles that we have on the planet today. I'll tell you about a discovery that we made of gut bacteria that co-diversify with host species. So I'll show you that many of our gut bacteria are derived and descended from these ancestral associations that have persisted within host species for millions of years, but that we humans have lost a lot of these ancestral symbionts. So I'll tell you some work we've done mapping these extinction events. And then in the third part, we'll shift gears a bit to tell you about some experimental work we've done in mice, looking at the consequences of disrupting these ancestral microbial associations for host phenotypes and fitness. When I got started working on these questions, we had a lot of data coming out from the Human Microbiome Project on humans. This is a many $million-dollar, tens of millions of dollars effort from the NIH to map the microbial communities that live in and on the human body, but we had almost no comparative data from other host species. So the first thing we did as evolutionary biologists is take this classic comparative approach. We reasoned that if we were to sample extant African apes in nature, we could use the known phylogenetic relationships among these host species to infer what the composition of the microbiome might have looked like in the ancestors of us and other hominid species. So it's a very simple comparative approach. So we worked with collaborators at the University of Pennsylvania and elsewhere, primatologist collaborators, to amass a large collection of fecal samples from African ape species residing in the wild in their ranges throughout equatorial Africa. So we have samples from chimpanzees and gorillas in Western Africa and Cameroon, Gabon. We have bonobos and gorillas in the DRC, the Republic of Congo, and also some eastern chimpanzees and Troglodytes strongypirthii from Tanzania, which are the famous Jane Goodall chimpanzees that you've probably heard about. And the first thing we did once we had these samples in hand was to profile the taxonomic composition of the microbial communities that resided within the guts of these hosts. So we have our microbiome samples, which in our case will be fecal samples that are generally representative of the microbial communities that reside in the large intestine. We extract DNA from the fecal samples, and initially we focused our analyses on this 16S rRNA gene, which is a gene that's present in all bacteria. But different bacterial groups have a slightly different sequence variant of this 16S rRNA gene. So it's an excellent marker gene for profiling the taxonomic diversity of microbes in a sample. So this is a very standard approach now in microbial ecology, the 16S rRNA-based amplicon sequence. So we extract DNA from our sample, we amplify this gene of interest, we sequence it, and then bioinformatically we can infer and decipher the relative abundances of different taxa in our sample. So at the end of the day, we get something like this. This is just a hypothetical toy plot to show you what these data can produce, where we can look at the relative abundance, we can estimate the relative abundances of taxa within these within our samples. So here I'm just showing hypothetical profiles for two samples, the different colors here representing the relative abundances of different bacterial taxa in these samples. Okay. So now I'll show you the results from some of our first efforts profiling the microbiomes of these apes. So I'll show you many of these stacked bar plots. These are what the data look like from gorillas. So here, each vertical line is a individual gorilla, and the different colors represent the relative abundances of different bacterial classes. So we're looking at a very high taxonomic level now at the composition of the gorilla microbiome. You can see hopefully that this is this beautiful kaleidoscopic diversity of different bacterial taxa that reside within the gorilla microbiome. These are what the data look like from bonobos and from chimpanzees. So right away, you can probably also see that there are big species differences between these two host species. Bonobos and chimpanzees look much more similar than either does to gorillas. I will also mention that we have represented here samples from chimpanzees that live together in sympatry with gorilla species. So the host species effects are maintained regardless of geography. Even when the chimps and gorillas live together, they maintain these distinct microbial communities. We were then able to compare our data with existing data sets at the time from humans, representing a range of lifestyles. So we have people living in small communities in the Amazon rainforest in Venezuela, people living in rural Malawi, and then many individuals sampled in cities throughout the United States. And we use the same methods that were used to generate these data, so we're able to compare across data sets. So what I hope you can see from this figure right away, which will be a main theme of the talk, is this greatly reduced overall levels of diversity in the human gut microbiota, particularly in these industrialized or urbanized settings. So the next thing we did as evolutionary biologists was to look at the rate of microbiome change over time and to ask whether the composition of the microbiome reflects host phylogenetic history. So I'm showing a very simple plot here of the divergence time between the host species that we sample on the x-axis and the dissimilarity of their microbiota taxonomic composition on the y-axis. So higher numbers mean more dissimilar microbiomes. You can see there's a nice roughly linear relationship between the degree of microbiome dissimilarity and the divergence of host species. So we have within species comparisons, bonobos versus chimps, which are just a couple of millimeters diverged. Bonobos and chimps are the genus Pan versus humans here, and then gorillas versus Pan and Homo sapiens here. So these results suggested there's been a roughly clock-like rate of microbiome turnover or microbiome change over the course of hominid diversification. But one thing that we noticed was that humans, especially humans living in cities in the United States, harbor gut microbial communities that are more divergent from those of African apes, wild living African apes, than we'd expect based on the evolutionary divergence between those humans and the African apes. So humans from cities in the United States always fall above this trend line. So there's been an accelerated rate of turnover in humans living in cities. This could be due to either a gang of new taxa or loss of taxa. And I'll just tell you that it turns out that the overwhelming pattern is loss of ancestral diversity. So if we look at the number of bacterial genera or species, families, it doesn't really matter what taxonomic level we look at in individual wild apes, gorillas, chimpanzees, bonobos, and compare those levels to the levels we observe in individual humans from our different populations. We always see lower levels of per individual microbial diversity in humans than in African apes. And this is most pronounced, again, in humans living in the United States in cities. And this result has since been replicated across many other human cohorts and other non-human primate studies. Yes?
This is diversity among classes of.
This plot is showing diversity of bacterial genera within individual hosts. I don't have it here, I believe, but this pattern holds regardless of taxonomic level of analysis. So we can look at species, strain, genus, family, class, order, phylum. At every level shows the same pattern. So this is showing an individual level view of diversity. So, number of taxa per host individual. In the next part of the talk, I'm going to zoom out to the population level and show you that there have actually been some wholesale losses, extinction events of some of these taxa from Homo sapiens as a species, and also from certain human populations. So I'll get into that in this part two, especially in this last part, the extinctions of ancestral symbionts from humans. So here I show you, there's a relationship between the community composition of the microbiome and the evolutionary history of the host species. We were next interested in what processes may be producing that association. So there are a couple of different non-mutually exclusive hypotheses that have been put forward in the literature to explain that pattern that we observed. This is taken from a nice review from Nancy Marie and Dan Sloan, published almost 10 years ago now in PLOS Biology. And cartoons of these two non-mutually exclusive processes are shown here. So on the left, or on your left, there's this process of co-diversification, which I would call an evolutionary process. I'll walk you through it. So here we have four kind of cartoon host species who are related to one another through this bold black phylogeny. And you'll note that more closely related species harbor more similar gut microbial communities than more distantly related species, just as we've seen in the African apes. And in this case, this pattern is produced by a process of co-diversification with gut symbionts, where the ancestor of these four host species harbored some set of gut microbial taxa that were then retained as the host species diversified. And those taxa then also diversified in parallel with their host species, leading to this parallel phylogenetic pattern. Every once in a while, a species maybe was gained or lost, but ultimately you end up with a state in the present day where more closely related hosts have more similar sets of gut bacteria. You could contrast that evolutionary process with what I would call an ecological process of habitat filtering, shown here on the right, where in this case, there are no long-term associations between individual gut bacterial lineages and their host species. Rather, host species simply sample from some common pool of environmental microbes, perhaps, some common pool of microbes. And because more closely related species are more similar in many traits, immunity, diet, etc., gut physiology, they select for more similar sets of gut bacteria. So this was sort of the next question we were hoping to get at. In other animal systems like insects, this process of co-diversification has been well studied, but at the time there were no examples of co-speciation or co-diversification between bacteria and mammal hosts. So we're interested, is that even possible? Our gut bacteria are acquired anew each generation. They're highly variable within us, respond to diet and lifestyle changes. We knew that already. So it wasn't totally clear that these relationships could be stable over such long evolutionary periods. So what we were interested in looking for is essentially this kind of pattern where we have the phylogeny of humans and African apes shown here on the left. Apologies to Jane Goodall for including a picture here. And the phylogeny of the bacteria on the right. And So this parallel phylogenetic pattern is indicative of an ancestral relationship that has maintained host specificity over millions of years. When we first tried to do this with the data we had at hand, these 16S data, we failed miserably. We realized very quickly that these data just didn't provide sufficient resolution to address the question. So 16S sequences evolve very, very slowly. Best estimates are that sequences diverge 1% per 30 million years, roughly. And so if you compare the sequences, homologous sequences from humans and chimps, for instance, there just simply hasn't been enough time for any sequence divergence to accumulate. There's not enough information there to build bacterial phylogenies or evolutionary trees over the time scale of hominid diversification. So I'll fast forward now 8 years, where we've been able to apply much higher resolution genome resolved metagenomic sequencing to profile the microbial diversity in chimpanzees and other primates and humans. These genome resolved methods really give us the phylogenetic signal that we need to test for co-diversification between gut symbionts and hosts. So in this project, we went back to our collection of chimpanzee and bonobo fecal samples and sequenced about 2,600 high-quality bacterial genomes directly from the fecal samples of these hosts. So we're able to sequence and assemble genomes directly from these metagenomic data. We then were able to combine our newly sequenced and assembled genomes with comparable genomic data from 22 other non-human primate species, which are shown here on the map, as well as 49 different human populations representing a range of lifestyles. So we have lifestyles including hunter-gatherers, people living in cities. I could talk more about all the lifestyles that are represented, but we have many, many different human lifestyles represented here. So ultimately, we were able to put together this phylogenetic tree of nearly 10,000 gut bacterial genomes assembled from humans and non-human primates. So you can think of this as like a compendium of all the gut bacterial genomes available from non-human primates, from humans. We were able to put them into a big tree that we could use to test this question of test for co-diversification, test whether bacteria co-diversified with their hosts. The next problem we ran into was once we have this tree, what do we do with it? How do we actually test for co-diversification given this giant phylogeny? So here I'm showing the phylogeny we constructed. Each tip represents a bacterial genome sequence. The different colors represent different bacterial phyla. You see we have over 10 phyla here. They're all the major phyla we expect to see are here. The branches in black down here represent divergence events between bacterial phyla that occurred 1 billion years ago, maybe even longer. And so these divergence events could not have been the product of co-diversification with host species. So we needed to find a way to look at the relevant parts of the tree that were coincident with the timescales of hominid diversification. So I'll quickly go over the approach that we took. Essentially, we were able to scan through the tree, looking only at the tips, the distal part of the tree, the time scales of the tree most recent, closest to the present, and test every single node in this appropriate timescale for co-diversification with host lineages. So I'm showing a little cartoon of this, how this works here. Here in black, we have the host tree, and we're literally, apologies again for the crude PowerPoint illustration here, but we're literally just scanning through the tree and testing every single node, one node at a time, for co-diversification with host species. And of course, we have to do many things to correct for false discovery rate. I won't bore you with the details. If you have questions, please let me know in the Q&A. I'll jump to the results, which is that we identified 219 clades, independent groups of bacteria, whose phylogenetic relationships are indicative of this ancestral association with hominid hosts and subsequent co-diversification with host species. Overall, this group of clades represented about 25% of the gut bacterial diversity we see in humans and non-human primates. So In other words, about 25% of primate gut bacterial diversity has arisen through this process of co-diversification with host species. So this was very exciting to us because it showed that these associations with gut bacteria in humans and non-human primates are evolutionarily ancient, that they're host species specific, and they've been maintained over millions of years of host speciation and evolution. So here are just a couple of examples of what these clades look like. So I'm showing here two different Prevotella species. This is a common genus of gut bacteria that each of us in this room have, I'm sure, within us. On the top are the bacterial phylogenies, and the bottom are the host phylogenies, and the dashed lines connect bacterial genomes from the host to the host from which they were recovered. You can see this nice parallel pattern indicating, 50 million year long association of between these gut bacteria and their hosts. Okay, so now we have this large set of gut bacterial clades that have co-diversified with hosts. We can be confident that they were in our ancestors with non-human primates and that they've maintained specificity to host species. We can then use these to test this question, ask this question: have humans lost any of these symbionts? To what extent have these ancestral symbionts gone extinct from human populations at the population level? And we can do that by looking for this characteristic phylogenetic pattern of extinction, which I've shown a cartoon here. Here we have a bacterial lineage on the right that's ancestral to humans and African apes. It existed as an ancestor of all extant African apes, but that has since gone extinct from the human lineage. So there's no representative in this tree of this clade from humans. This is sort of the classic pattern of symbiont extinction that we would expect to see. So in total, we identified 129 clades of co-diversifying symbionts that we could infer to be ancestral to the hominin, to humans and chimpanzees and bonobos. Nearly half, 45% of those ancestral clades were absent from all human populations that we sampled, regardless of lifestyle, including hunter-gatherers, agriculturalists, many geographic regions represented. None of them contained any of these 45% of these ancestral symbiont clades. So here I'm showing the phylogeny again, just turned on its side on the left, and the horizontal bars here mark ancestral clades that have gone extinct from Homo sapiens. That's a hypothesis. It could easily be disproven. If someone finds one of these clades in any human, we could change this figure and it would be updated. But as of now, we think they really are extinct from humans. When we look just at industrialized populations, we see that about 55% of these ancestral symbionts have gone extinct from humans. So there are some taxa that are found in non-industrialized human populations, but that are absent from human populations. Shown here. So what does this mean? Overall, I think we can conclude from this that the story that we could tell from the data is that there have been 2 rounds of microbiota extinctions from the human lineage. So the first likely predates the diversification of extant human populations. It occurred before our most recent common ancestors of all of us. It may have been driven by dietary changes that were known to have occurred during human evolution, such as the transition away from eating highly fibrous foods that are known to promote microbial diversity towards cooking food and eating meat. Then there's been a second loss of microbial diversity, extinction of these ancestral symbionts that's specific to industrialized human populations. And these are the ones that maybe are driven by these recent lifestyle changes, antibiotics, hygiene practices, et cetera. We can also do the complementary analysis just as a sanity check, looking at extinction rates from chimpanzees and bonobos. And the take home there is that we see despite a 10-fold greater sampling effort from humans, we see a roughly 6-fold greater rate of extinction from humans than from chimpanzees or bonobos. So it really does seem like humans are special and in the sense that we've experienced these accelerated rates of microbiota extinctions. So I want to just give you a picture of what some of these look like. So here, I'll just show individual clades that are, zoomed in versions of parts of this tree here. So here's a Lacnosporiaceae species that is detected in gorillas, chimpanzees, and multiple subspecies of chimpanzees and bonobos, that is completely absent from all human populations. The Bacteroideales species, another Prevotella species, Oscillosporiaceae, and many different bacterial groups, Fibobacter. So this pattern of extinction spans the entire bacterial phylogeny showing on the left. Another point I wanted to make is that we observed very little evidence of extinction from humans of these non-codiversifying clades that do not show a pattern of co-phylogeny with their host species. So we could look at clades that showed no evidence of codiversification, but were present in chimpanzees, bonobos, and gorillas. Of those, almost all of them were also present in humans. Over 97% of those were detected in humans. So this tells us that there's something special about our host species specific co-diversifying gut bacteria that makes them particularly susceptible to these accelerations of extinction rates. One conclusion I was hoping to make here, and hopefully you can take with you, is that the loss of our ancestral microbial diversity has really been centered around these co-diversifying host species specific clades. These are bacteria that only exist within us. Our specific lineages have co-diversified with us. Once they're lost from us, there's no reservoir from which we can regain them. Once they're lost, they may be lost for good. Whereas these non-host specific gut bacteria, these non-co-diversifying clades, which include things like environmentally derived taxa, E. coli, staph, etc., those can be retained quite easily because if they're lost from any population, they can be regained through the environment. Okay. So in the third part, I want to shift again to talk about some experimental work we've done in mouse models to ask what are the consequences of disrupting these ancestral symbioses for hosts. So some of you may have been exposed to this hypothesis, the hygiene hypothesis that has been popularized over the past decades. A recent iteration of it is focused on the microbiome. So this is a book written by Marty Blazer just down the road at Rutgers called Missing Microbes, How the Overuse of Antibiotics is Fueling Our Modern Plagues. And the thesis of the book is basically that humans, particularly in industrialized countries that are overusing antibiotics, are driving extinctions of these ancestral microbes, and that's having a deleterious effect on our immune development, our metabolic health, and so on. That is giving rise to these modern diseases. So there have been many opinion and review articles written about this idea, but I would argue very little concrete data backing up the idea that these losses in ancestral microbial diversity are deleterious for humans. So this is the question I'm interested in talking about for the next 15 minutes or so. So as of now, I will make no strong claims about whether these are deleterious or neutral or beneficial. It's very difficult to do experiments in humans to test the consequences of the losses of these ancestral microbes. But we're lucky in the sense that rodents and their gut microbiota have also co-diversified with one another. So we've done a similar study looking at rodent species, including these species shown here on the top left, finding that, again, about 25% of the gut bacterial diversity in rodents has arisen through co-diversification with host species. And here on the bottom, I'm just showing a few examples of these symbionts that show an ancestral relationship with host species and co-diversified with their host species. So we can actually, I think, use rodents as a model to start to investigate what are the consequences of disrupting these ancestral associations for hosts. So my lab has been developing these wild-derived rodent models as an experimental system for studying questions about co-adaptation between the microbiota and hosts. To what extent have microbes affected the evolutionary adaptations of their hosts? And conversely, how have associations with hosts shaped the adaptive evolution of the microbes? Unlike humans, we can rear these at the lab, we can rear them under germ-free conditions, and we can do manipulative experiments. So in our lab, we've had all these species shown here on the left at one time or another in a common garden environment. We've shown that they're able to maintain their host species specific gut microbial communities in this common garden environment, and we can use them for experiments. So one of the nice things that we can do with rodents is rear them germ-free. So this is showing a non-abiotic isolator in which there are several cages of mice that have been born and reared in the absence of any kind of microbial exposure. So they're reared in a totally sterile environment. They have no exposure to bacteria whatsoever.
Just a clarification question.
Yeah.
How do you deliver them through C-section to?
Yeah, so once you have a germ-free mouse, you can make more fairly easily. The first germ-free mouse was derived by excising the embryonic sac from the mouse, disinfecting the embryonic sac, and then hand rearing the pups in a sterile isolator under sterile conditions. So literally, someone pipetting daily every few minutes. And so
you just have offspring of that.
And then once you have a germ-free line going, you can derive other genotypes and lines germ-free through embryo transfer to that germ-free mother. So in utero, mammals are sterile essentially. And so then that way you can derive additional lines. So the hard part is getting the first one. Once you have one, you can maintain. Yes.
Is there contamination, right?
I'm sorry, could you repeat?
Is there any contamination from food?
Yeah, so the food is heat shocked, UV sterilized. And these germ-free facilities must engage in constant monitoring for contamination, and it is a problem. So there are cases where an isolator will get contaminated, an experiment will be ruined, and you have to start over. So it's a tricky job, but it's one that people have really worked out. I mean, there are now core facilities that do this, full time as their run these facilities as a service. Yeah, good questions though. Okay, so the nice thing you can do with these germ-free mice is introduce defined sets of microbes. You can really do manipulative experiments in a very controlled way to test the effects of different combinations of microbes on the host. So I'll tell you just really quickly about a couple of experiments we've done recently using these germ-free mouse systems. So the first thing we were interested in looking at was whether the bacteria that are specific to different host lineages have adapted to those host lineages. And so we tested for this through competition experiments in germ-free mice, where we took germ-free house mice, Musculus domesticus, and inoculated them with either native or non-native microbiota. And by native microbiota, I mean microbiota from house mice, Musculus domesticus, and by non-native microbiota, I mean microbiota from other species of rodents, including other mouse species, as well as more distantly related species, Paramyscus maniculatus. And the prediction is that if microbes are locally adapted to their host species, we should see the Mus domesticus microbes outcompeting the non-native microbes in these germ-free mice. And that's what we see over and over again. We've done many of these experiments now. I'm just showing one result here. On the y-axis, I'm showing this ratio of native to non-native microbes. 0 would indicate a 50-50 mix between native and non-native microbes. On the x-axis, I'm showing the sampling points of the germ-free, ex-germ-free mice, the non-abiotic mice after the colonization. So you can see in every case, we see a competitive advantage for the native microbes over the non-native microbes. Here, the different shapes and colors represent different combinations of native and non-native microbes. So there does seem to be local adaptation of these microbes to their host species. We've also done experiments looking at the other side of the coin, looking at whether host species have evolved dependence on their specific microbial communities. And so to do this, we're able to take again, microbiota from different host species, from house mice, Mospredis, or distant relative Mospahari, put them into germ-free mice and look at the effects of those different microbiota transplants on the germ-free mouse's phenotypes. The expectation being that fitness-related traits should be sort of maximized when you have your native microbes and negatively affected relatively by these non-native microbiota. And we've seen some evidence for that, exactly that. So here I'm showing full change of body weight, just the growth rate of these germ-free mice over the first 10 weeks of life. And then here each line represents a different mouse that was given either native or non-native microbiota. And the colors represent which microbiota the mouse was given. So blue represents mice that were given a native Domesticus microbiota, yellow, the microbiota of a close relative Mospredis, and green, the microbiota of a more distant relative Mospahari. The data broken down by females and males. And you can see there's this gradual reduction in growth rate as you give the mice a microbiota from more and more distantly related hosts. Exactly what you'd expect if hosts have evolved some dependence on their native microbes. Here, the dashed line is just the growth rate of germ-free mice. So overall, we see germ-free mice grow slightly slower than mice that receive their native microbiota. One thing that's interesting to us from this experiment was that mice given the non-native microbiota from Mospahari, a very distant relative, actually grew worse than germ-free mice. So in some sense, it may actually be worse to have the wrong microbiota than to have no microbiota whatsoever. We also see in these mice harboring non-native microbiota upregulation of various markers of inflammation. So one of them, shown here on the y-axis, is the abundance of this macrophage inflammatory protein 1 beta in the blood serum of these mice. This became kind of involved in recruiting macrophages to sites of inflammation. You see it very low in germ-free mice, slightly elevated in mice that have their native microbiota, but elevated even more to sort of an abnormal level in mice that receive a non-native Spreadis or Bahari microbiota. So there's some consequences here of disturbing or replacing these native co-diversified gut microbial communities. So we then more recently have been interested in looking at laboratory mice and to what extent they've retained their native wild mouse microbes. And it turns out that if you look at laboratory mice lines, which have been in the lab for over a century now, they were first derived early 1900s and inbred ever since in these very artificial conditions, like humans, they have lost about half of their ancestral microbes. So here I'm showing a cartoon of the bacterial phylogeny from rodents. Each colored circle here on the phylogeny represents a clade of bacteria that is co-diversified with rodent hosts. And the top bar here is showing whether that clade is present or absent in lab mice. So the absence of a circle here indicates the absence of that ancestral clade from lab mice. So these are instances of extinction of ancestral symbionts from lab mice. And it turns out if we look at which taxa are lost from lab mice, we see a sort of striking parallel between the taxa lost from lab mice and the taxa that are lost from humans. So one example is Trypanema. So here I'm showing a phylogeny of Trypanema gut bacterial strains derived from different rodent host species shown on the right. And if you look closely at the species labels, you'll note there's the absence of any lab mouse derived strains here. We also see this group Trypanema being lost from human populations. So one thing I'm very excited about now is, can we use these parallel losses of the same taxa ancestral symbionts from humans and mice to design some experiments in mice that maybe could point us in the right direction about what the consequences of these losses may be for humans. So I think there's the potential here to use what's happened in lab mice as sort of a model for understanding what's happened in humans in terms of gut bacterial extinctions. Here I just have a picture of another Trypanema, different species, but these are, in addition to being mutualists in the gut, can sometimes cause disease, syphilis. So to end, I'll give you a teaser about a project that's still underway in in my group, led by a EB graduate student, Abby Landers, who's a fantastic graduate student in my lab, where she is testing whether we can restore the microbiota of laboratory mice using fecal microbiota transplants from wild mice, and whether those FMTs and the subsequent restoration of the microbiota can sort of rewild the immune phenotypes of these lab mice. So in her experiment, she's taken microbiota samples, fecal samples from wild mice, and gavaged them, just pipetted into the mouth of lab mice, and let those mice give birth to an offspring generation. And then she's comparing both the microbiota and the immune profiles of these lab mice that receive a wild-derived FMT with lab mice that receive a control FMT, which is either an autologous gavage, an FMT of a lab mouse microbiota into the lab mouse itself, or just a negative control, a PBS negative control gavage. So we've been able to show that the gavage works. We're actually able to restore some of the wild microbiota diversity into lab mice just through a single gavage, which I'm showing here. This is the percent reads mapped to the wild microbiota and the control FMT here at the wild. We see much higher rates of mapping, meaning more colonization of wild taxa in the mice that receive the wild FMT compared to the control. We're also able to show that this is transmissible to the next generation. So once you introduce these wild microbes to house mice, they can be maintained over generations in those mouse lines, which is a nice thing. And she's also looking at the immune consequences of the loss of these symbionts or the reacquisition of them. And these are very preliminary results, but I want to share one result that's just very fresh that I'm excited about, showing that the offspring of mice that received a wild fecal microbiota transplantation shows some immune changes that make them look more like wild mice than lab mice that receive the control FMT, or just lab mice that receive no FMT whatsoever. So this is showing the proportion of cells in the blood belonging to the cell type effector memory CD4 positive T cells. So these are these memory CD4 positive T cells that have had some antigen exposure in the past. And this pattern we see in the wild FMT mice very much resembles the pattern we see in wild mice. So the inference from this is that these lab mouse lines have immune deficits due to the loss of their ancestral microbiota that can be restored through this FMT. Another interesting feature of this, I think, is to note is that we don't see this effect in the parent generation. The mice that were actually given the FMT show no immune differences. It's only in the offspring of those mice that the immune differences emerge, which I think implies the existence of some critical windows where these microbes may be important for immune training. It may be that exposure in early life is what matters. Okay, so I think hopefully right on time. There's about 15 minutes left for questions. Thank you for sticking with me. I know this is a bit of a departure from the maybe typical talk given in the seminar series. If there are just a few take-homes that you leave with, I want them to be this. So our gut bacteria are not just a haphazard collection of microbes. They're in us. There are in us bacterial lineages that have co-diversified and likely co-evolved with us for millions of years. That we are in the middle of a bout of microbiota extinction in humans, and this is especially pronounced in populations in industrialized settings. And I was agnostic at first about whether these extinctions are good or bad or neutral. I'm starting to agree with Marty and the Sonnenbergs at Stanford and other people who have been beating the drum that these losses of ancestral microbiome may actually come at some cost of our immune development. And so with that, I will stop here and just acknowledge all the various collaborators. This has really been a collaboration between many fields: mammalogy, primatology, evolutionary biology, genetics, microbiology. There are more people than I can fit on the slide, and also thank the funders. So thank you.